Good morning all. Today we are going to see the industrial production of glutamic acid. What is glutamic acid? Glutamic acid is a amino acid. You know there are 20 different types of amino acids, and among those 20 different types of amino acid, glutamic acid is one of them. Glutamic acid is one of the first amino acid to be industrially produced, and it was produced by 17 companies, and 13 of the 17 major manufacturers of amino acid production industries were located in Southeast Asia, and Japan, Korea, Taiwan. They were the foremost leading companies which produced the amino acids. The taste enhancing property of glutamic acid was first discovered in 1908 in Japan, and the commercial production of sodium glutamate from acid hydrolysate of wheat and soy protein began soon after. So, in 1957, the L-glutamic acid was discovered as a product in the spent medium of Corynebacterium glutamicum. So, this Corynebacterium glutamicum is the microorganism which is used for the production of glutamic acid, that is the amino acid. Later, L-lysine was produced commercially, and in 1985, that is the world production of biochemically produced amino acid was 460,000 tons, and of these 460,000 tons of amino acid produced, 370,000 tons per year was sodium glutamate. Means the sodium glutamate was produced in huge amount, and only 70,000 tons of L-lysine was produced. But because of the anticipated increase in demand all around the world led to the increase in production of these amino acids, and this amino acid was produced commercially. Amino acids have extensive industrial application in different industries. About 66% of amino acid being produced is used in the food industry. 31% is used as a feed additives for purpose in animal feed, and in 4% is used in medicines and cosmetics, as well as the starting material in the chemical industry. So, in the food industry, amino acids are used alone or in combination to enhance the flavors. So, monosodium glutamate, as you know, it is used to increase the flavor, and it is very much liked by its taste is very much liked, and it is used in many spicy recipes like Manchurian and the Chinese Hakka noodles, or in the even in the Maggi also. Last few years ago there was a controversy that the percentage of monosodium glutamate was higher, and therefore that company has to face some legal actions or something like that. So, monosodium glutamate or the sodium glutamate is basically a that is the flavor enhancer, and therefore it is used in many of the food industries. Then you have other amino acids also, like L-cysteine. It improves the quality of the bread, and it was usually used in the baking processes in Japan. L-tryptophan, when it is combined with the L-histidine, it acts as an antioxidant and is used to keep powdered milk from getting rancid. So, powdered milk usually it gets rancid, or you have a buttery appearance or the taste, and that it can be avoided when you are using the L-tryptophan and L-histidine. The aspartame, it is a sweetener, particularly the diabetic patients use those sweeteners. So, this aspartame is made from the L-phenylalanine and L-aspartic acid. So, it is a low calorie, and usually it is having a low calorie, means not having so much of energy, and therefore it is used in the soft drinks. You have many different types of nutritive food products which are derived from plants, but usually the plant proteins, if you see, that is the protein powders which are usually available in markets, which are derived from only plant based. These protein powders, they are deficient in certain types of essential amino acids like L-lysine, L-methionine, or L-threonine, or L-tryptophan. Then usually this particular amino acids or the essential amino acids has to be supplemented from the outside, and therefore commercial production of amino acid is very necessary in order to make that particular or in order to add the nutritive value to those plant based products. So, in view of the scarcity of the nutritious food during or in the third worlds like Africans, or even India is also included in the third. So, with the ever increasing need to supplement all those that is the people, or you have to feed them, and you have to feed the or you have to give the food enriched with the different types of essential amino acids, and therefore you have to commercially produce these particular types of amino acids at the industrial level, and therefore promising amino acid producing industries came forth and started producing the amino acids. So, amino acids, they are playing an important role in biology of life, in biochemistry as well as in the industrial chemistry. Since the amino acids, they are the building blocks of proteins, they play an essential role in the metabolism of living organism as well as in the regulatory mechanism of the different processes taking place in the organism. Large scale chemical and microbial production processes, they have been commercialized. We can usually produce amino acid commercially, either by chemical method also, or by microbial production, that is by means of fermentation using different types of microorganisms, or you can by the third method also you can produce the amino acid, that is by hydrolysis of the proteins. So, these are the three methods by which you can obtain the amino acid. Current interest is developing because peptide derived chemotherapeutic agents have been used in treatment of different types of diseases or the ailments. It has heightened the importance of rare and non-proteogenic pure amino acids. So, because of this the pure amino acid has gained its importance. So, this is the structure of primary structure of protein. So, this protein is having different types of amino acids, and these amino acids are linked through peptide bonds, that is the CONH bond. So, this is the structure of a primary structure of, that is protein molecule. This is the alpha structure, or you can call it as a helix structure, and this is the beta sheet. So, you have the arrangement of the or you have the protein structures in different conformations, that is the primary structure, secondary structure, and the tertiary structure. So, as well as the quaternary structure of protein. So, this is the secondary structure, alpha helix structure, and this is your primary structure, this is your secondary structure, and when the that is the polypeptides are changed in a particular three-dimensional structures, that is in the form of a beta sheets, which can be a anti-parallel or it can be a parallel, then you get a tertiary structure. So the importance of amino acids, they are versatile chiral, that is they are optically active, building blocks of the whole range of fine chemicals. So you have many different types of chemicals which you can have it from the amino acids. Then amino acids are therefore important as nutrients, foods, seasonings, flavoring agents, starting material for pharmaceuticals. Even the amino acids are used in cosmetics because amino acids are acting as a oxidants and other chemicals also. So amino acids can be produced by chemical synthesis, or it can be isolated from the natural materials, or you can produce it by the fermentation process by using different types of microorganisms, or it can be a chemoenzymic method, what I have said earlier, that is if you hydrolyze the protein and then you can extract your particular amino acid. So this by means of your enzyme action you can have that particular amino acids. Now we are come to the production of glutamic acid. Now what is glutamic acid? Glutamic acid is a alpha amino acid and used in biosynthesis of the proteins. So it contains alpha amino groups, and this is usually a protonated form, that is NH2 group is protonated to form the NH3 positive, and it has the alpha carboxylic acid which is deprotonated, means you will have COO negative, and it has a side chain carboxylic acid. Since it is a acid, glutamic acid, it is containing the another COOH group. Polar negatively charged at physiological pH, and it is a aliphatic amino acid. Since see here, this is the structure of glutamic acid, so it is having a aliphatic chain, and therefore it is called as the aliphatic amino acid. It is non-essential in human, means the body is having the capability to synthesize it. So for humans there is no need to have the glutamic acid in from outside, because the human body itself synthesizes the glutamic acid. So this is the structure of glutamic acid, C double bond O and OH, this is the extra O COO group. Okay. So what are the applications of glutamic acid? It is used for food production. In food production, how they are used? They are used as a flavor enhancer to improve the flavor. They are also used as a nutritional supplement, because their certain essential amino acids are not satisfied in the plant products and they have to be added. Then in the beverage also they are acting as a flavor enhancers. So in soft drinks and wines they are used. In cosmetics they are used as a hair restorer. So if you want to treatment the hair loss or falling of the hairs, then you can use this glutamic acid. And it is also used in the anti-aging creams also, to in order to prevent the wrinkles or to prevent the aging. And in agricultural and animal feed also it is used as a nutritional supplement, and in feed additives in order to enhance the nutrition. And other industries also it is used as an intermediate for manufacturing different types of organic chemicals. So now we will see the biosynthesis of glutamic acid. So amino acid precursor is converted to the target amino acid by using one or the second enzyme. So I will explain you with the next slide. First of all, in this slide we will see what are the different enzymes which are responsible for converting the intermediates into the glutamic acid or the glutamine. So it allows the conversion of a specific amino acid with the microbial growth, thus eliminating the long process of from glucose. So what it is doing? It converts the specific amino acid without microbial growth, and therefore instead of undergoing a long metabolic pathway starting from the glucose, you can directly convert a specific amino acid into the glutamic acid also. So glutamine can be converted into glutamic acid. Raw materials for the enzymatic steps are supplied by the chemical synthesis. So, whatever the material is required, it is usually supplied by the chemical synthesis, and the enzyme itself is either isolated or whole cell form can be used for the microbial fermentations for the production of glutamic acid. So, what are the reactants? So, these are the reactants from glutamine in presence of water. You can get glutamic acid plus ammonia, and N-acetyl glucosamine in presence of water you will get glutamic acid and acetate. Alpha ketoglutarate in presence of reducing agent NADPH plus ammonia, you will get glutamic acid plus oxidized form of NADP and water. And alpha ketoglutarate in presence of alpha amino acid you will get glutamic acid and alpha oxoacid. And 1-pyrroline 5-carboxylate in presence of oxidized NAD positive will give you glutamic acid plus NADH and water. Here water is added, and N-formimino L-glutamate in presence of tetrahydrofolate you will get glutamic acid, and 5-formimino tetrahydrofolate, that is folate. So, these are the particular enzymes which are catalyzing these reactions, and you will get the product as a glutamic acid. So, now we will come to the industrial production of glutamic acid, and what is the organism being used for its for industrial production. So, usually the microorganisms, they typically grow on large scale and then they produce the products by undergoing that the products are produced either by the chemical transformations also. So, glutamic acid is produced through the fermentation process, and the major microorganism which is used is Corynebacterium glutamicum. Since it is a glutamic acid, it is called as the glutamicum. So, Corynebacterium glutamicum. So, the classic methods are used to select the high yielding microbial variants, as we have seen what are the different strain improvements like the induced mutations or by recombination or by genetic engineering. You can yield the or you can increase the yield of your product using that particular genetically recombinant or the that is by means of site directed mutagenesis carried out. You can improve the yield of your amino acid being produced by that particular organism. Then the properties of the useful industrial microbe, it includes that this Corynebacterium produces the spores, and that can be easily inoculated, and they grow rapidly on the large scale in inexpensive medium, and it produces the desired product very quickly. And usually when you are using a microorganism, you should have one important thing in mind, that it should not be a pathogenic microorganism, and it should be amenable to the genetic manipulations, because if you are doing a genetic manipulations, then you can improve that particular strain, and you can increase your yield. So, these are the properties when using industrial microorganism for the production of amino acids. So, now we will see the industrial production of glutamic acid. So, the manufacturing process of glutamic acid fermentation is usually comprising the fermentation, that is the fermentation media, that is formulation of media, what substrates are being used for the amino acid production, then what is the how the inoculum is added and how particularly the fermentation product is carried out, and later on the purification processes. So, there are 4 types of fermentations which are used for the production of glutamic acid, that is the batch fermentation, fed batch fermentation and continuous fermentation. Now, we will see the batch fermentation. So, batch fermentation means once the fermentation media is added along with the inoculum or the starter culture, then the incubation is carried out till the fermentation is over, or you form a product, and after the formation of product only that particular recovery of fermentation product is usually done. But in a continuous fermentation, usually what happens, the fermentation media is being added and continuously the product is being recovered. So now in the batch fermentation, usually it is the widely used for the production of most of the amino acids, and it is a closed culture system which contains the initial and limited amount of nutrients, because once the nutrient is added, again you don't add the nutrient. This particularly for one particular fermentation process, only one time nutrient medium and the starter culture is added. A short adaptation time is usually necessary, because the cells usually adapt themselves to that particular substrates, as well as they make their own, they make the enzymes responsible for the cell growth, and the cell enters, then the cell enters the logarithmic or the exponential growth phase. But later on what happens, the nutrient soon becomes limited and they enter the stationary phase, in which the growth almost ceases. And in glutamic acid fermentation, production of the acid normally starts in the early logarithmic phase. See here, when does the glutamic acid fermentation start? It starts in the early logarithmic phase and continues through the stationary phase. So till the stationary phase it continues. For economical reasons only, the fermentation time should be as short as possible in order to have a high yield of your glutamic acid at the end. A second reason not to continue the fermentation till the late stationary phase. You cannot continue till late stationary phase, because there is a problem of contaminant product formation, because along with the glutamic acid you can get any other products besides the glutamic acids. So the lag phase can be shortened by using the higher concentration of the seed inoculum. If you increase the inoculum or the starter culture, then the log phase can be shortened, and the seed is produced by growing the production strain in flask and then in the smaller fermenters. So this is your batch fermenter. See here, this fermenter, it is equipped with all sorts of accessory equipments to know the pressure, to maintain the temperature, and inoculum addition, then for outlet for the fermentation media, inlet for the, see here you have the pressure gauge, then you have the sterile air passage, okay, entry of sterile air, everything is there. So then you have the cooling jackets inside, and the coolant is usually used. So this is the typical fermenter which is used for the amino acid production. Fed batch fermentation. So batch fermentations which are fed continuously or intermittently. Sometimes what happens, you you feed the fermenter with the nutrient medium. So in this way the volume of the culture increases with the time. So usually what happens, initially it was at this level, the fermentation media was at this level, but the nutrient, but after a period of time that microorganism has utilized all the substrate, and therefore there is now necessity to add the nutrient medium. So the nutrient medium is added intermittently, and now the volume has increased here. Then again after a period of time, that substrate is converted into the glutamic acid, and then there is a limitation of that particular medium, and so the feed, that is the nutrient medium, has to again added, and now the volume has increased. So there will be increase in the volume with respect to the time. The residual substrate concentration may be maintained at very low level, and this may result in removal of catabolite repressor effects and avoidance of the toxic effects of the media components. So by doing so, what happens, if you are adding the if you are adding the nutrient media, or you are adding certain catabolite, then they might have a suppressing activity and you will not get the proper yield. So, when they are added intermittently, then there is no catabolite repressive that is repressive effects, and therefore the toxic effect of that particular medium component can be means kept in a low concentration, or the you can avoid that particular toxic effect. Then there is a oxygen balance, because since it is a aerobic process, this aerobic fermentation, you have to maintain the sterile air, and the feed rate of the carbon source, usually it is used to regulate the cell growth rate and the oxygen limitation, especially when oxygen demand is high in the exponential growth phase. So, when the microorganism is growing in the exponential growth phase, then usually the oxygen demand by that microorganism is very high. So, you have to keep the oxygen rate in a particular state, and that has to be satisfied when the cells are growing exponentially. In the continuous fermentation, usually it is a open system. The sterile medium, nutrient medium is added to the bioreactor continuously, and usually the fermentation media is withdrawn. So, an equivalent amount of converted nutrient solution with microorganism is simultaneously removed from the system. So, see here, these are the four nutrient media, four fermenters which are in closely associated with one another. So, here what you are doing, you are adding the nutrient media, as well as you are removing the fermentation media. So, again it is, if you think that the fermentation is not done properly, then again it is recycled and it is put in this fermentation. So, from here it is going to here, and then from here it is going to here, and then from here it is going to here. So, it is in a continuous process. So, this is how the continuous fermentation takes place. Industrial production of glutamic acid is usually carried out by using a substrate, which is the sugar cane. So, sugar cane is squeezed to make the molasses, and then the heat sterile raw material and other nutrients are put in the tank, and then the microorganism, that is Corynebacterium glutamicum, is added to the fermentation broth as a inoculum, and this microorganism converts the sugar to produce the glutamic acid. So, the fermentation broth is acidified and the glutamic acid is then crystallized. So, now we will see we see the biosynthesis of glutamic acid. So, how the glutamic acid is synthesized using glucose as the carbon source. So, as you know, the glucose is converted first to glucose 6 phosphate, and then the glucose 6 phosphate is converted to pentose 5 phosphate by removal of carbon dioxide, and then this pentose 5 phosphate is converted to acetyl CoA, and then this is the citrate. Okay, this is your Krebs cycle, this is your glyoxylate, as I have already told you. Then this is your from acetyl CoA you can directly lead to malate in presence of glyoxylate, and this is the isocitrate which can be converted to glyoxylate. Okay, as I have told you in the citric acid cycle. Now, this isocitrate is converted to alpha ketoglutarate. Okay. Now, in presence of isocitrate dehydrogenase enzyme, this isocitrate is converted to alpha ketoglutarate, and this alpha ketoglutarate in presence of enzyme that is glutamic acid dehydrogenase is converted to glutamate. So, this is converted to glutamate. Alpha ketoglutarate, in presence of glutamic acid dehydrogenase enzyme, it is converted to glutamate. Then from this glutamate you can get the N-acetyl glutamate, and this N-acetyl glutamate is synthesized in presence of glutamine that is N-acetyl glutamate synthase. Now, this from glutamate also you can synthesize the glutamine, and this glutamine is synthesized in presence of the enzyme glutamine synthetase. So, it requires the ammonia, and ATP hydrolysis takes place, and therefore, usually you will get the glutamine. So, this is the process where the glutamate is synthesized, and from glutamic acid you can get the glutamine. So, this is how the biosynthesis of glutamic acid takes place. Microorganisms also use another pathway in which instead of glucose they can use the acetate. So, acetate can be used as a carbon source. So, acetate is first converted to acetyl phosphate, and then from acetyl phosphate you get acetyl CoA, and this acetyl CoA, in presence of oxaloacetate you get the citrate, and then from citrate cis-aconitate, then isocitrate, and then from isocitrate you get the alpha ketoglutarate, and from alpha ketoglutarate you get the glutamate. So, glutamate can be synthesized from acetate also, because acetate or acetic acid can be utilized as a carbon source. Glutamic acid can also be produced by using the strains, microbial strains like Brevibacterium, Microbacterium or Arthrobacter, and but basically the industrial production of glutamic acid is carried out by Corynebacterium glutamicum. During the glutamic acid production, usually the excretion of the glutamic acid is carried out by increasing the cell permeability. Now, what is this cell permeability? Usually any type of microorganism, they have the cell wall, and cell wall is made up of the peptidoglycan layer, and usually the porosity of the cell wall is is of particular importance, because when you are increasing the porosity, then the excretion of the glutamic acid can take place smoothly, and you can have the that is glutamic acid accumulation or the excretion. So, usually you can increase by biotin deficiency, means if you want to increase the permeability of glutamic acid bacteria, then you can obtain it through biotin deficiency or through oleic acid deficiency in oleic acid auxotrophs, or you can usually you can add saturated fatty acids or fatty acid derivatives, or by addition of penicillin also. You can add penicillin also in order to increase the porosity, and by increasing the porosity you can usually excrete out the glutamic acid into the fermentation medium. So, all these glutamic acid producing strains, they have a growth requirement for biotin. Means if you want to grow the Corynebacterium glutamicum in good amount or in in excess amount, then biotin is a factor. But because it is a essential coenzyme in fatty acid synthesis, so it is a requirement for the growth. But once the growth has been attained and the glutamic acid has been produced, then you can just limit the biotin, means you do not add the biotin, and because of the deficiency in the biotin, then the porosity of the bacterial cell wall increase, and the glutamic acid can be excreted out. In the presence of biotin concentrations greater than 5 microgram per liter, increased oleic acid synthesis takes place, which results in the high phospholipid content of the cell membranes, and because of the high lipid content, usually the bacterial cells are incapable of excreting glutamic acid, and up to 25 to 35 microgram L-glutamic acid per mg dry weight can be accumulated within the cells. Or you can say it as that they are accumulated intracellularly, so that biosynthesis is stopped because of the feedback inhibition. And when the biotin is limited, or when there is a shortage of biotin in the fermentation media, then it causes the membrane damage, and usually because of this, because of the reduction of the phospholipid synthesis, it usually what happens, the glutamic acid is excreted out. Similarly, the addition of penicillin in the logarithmic growth phase, it promotes the significant excretion of L-glutamic acid, even if there is a presence of biotin. Means even biotin is present, but if you are adding penicillin in the logarithmic phase, then the glutamic acid can be excreted out. Because of the penicillin use only, it has been a favorable or economically feasible fermentation process, because we can use cane molasses or the sugar molasses as a substrate for the glutamic acid fermentation. The fermentation conditions, usually the glutamic acid production is optimally carried out, and it converts, that is the bacteria converts, Corynebacterium glutamicum converts 50 to 60% of carbon source to L-glutamic acid. If favorable conditions are not present in the fermentation process, then usually there will be a lower yield of glutamic acid. Usually, what are the different carbon sources used in the fermentation media? A wide variety of carbohydrates are used as the carbon source in the fermentation. Among them are the monosaccharides, the glucose and the sucrose, which are frequently used. However, you can use the fructose, maltose, ribose, xylose, etc. But a more efficient way of carbon source which is used, which is economically feasible also, that is your cane molasses or the beet molasses. But since this cane molasses or beet molasses contains high biotin content, usually penicillin is used. Penicillin or fatty acid derivatives like twin 60 is used to the fermentation process, and this results in the higher yield of L-glutamic acid. Processes utilizing other carbon sources like methanol, ethanol, acetaldehyde or N-alkane have also been developed, but the cost effectiveness of this process depends largely on the price of the substrates like methanol or ethanol or acetaldehyde being used, and therefore it is not a economically feasible process. Means if you have to carry out the L-glutamic acid production using this particular carbohydrate sources, then it is very expensive when compared to the fermentation processes which utilizes the beet molasses or the cane molasses. In addition to the carbon source, nitrogen sources are very important. That is ammonium salts, ammonia in the form of gaseous or in the aqueous solution, they are also used as the nitrogen source. So the pH control is usually that is when ammonia is used, then usually the pH control has to be seen effectively, because it also obviates the problem of ammonia toxicity, because microorganisms, they are having a detrimental effect on their growth when the fermentation media is having excess of ammonia. Growth factors, the optimum biotin concentration, it depends on the carbon which is being used. If you are using a 10% glucose, then the biotin added should be 5 microgram per liter of the media. But if you are using a lower glucose concentration, then naturally the biotin concentration should be lower too. If you are using a acetate as the carbon source, then the biotin content should be 0.2 to 1 microgram per liter. So, depending on what type of carbon source you are using for the fermentation media formulation, usually you have to add the biotin component. Oxygen supply. So, optimum glutamic acid yields are obtained when 3.5 into 10 to the power of minus 6 mole of oxygen is given or provided to the fermentation media. So, oxygen concentration should neither be too low nor it should be too high, because under oxygen deficiency excretion of lactate and succinate occurs, but when there is excess of oxygen, usually in presence of ammonium ion deficiency, the growth ceases, and the production of alpha ketoglutarate also ceases, and therefore glutamic acid yield is very low. The fermentation is carried out at 32 to 33 degrees centigrade, and the pH is maintained at is set at near about 8.5 with ammonia, and usually during the fermentation it is usually 7.8. The fermentation is stopped after 30 hours, and glutamic acid is the yield of glutamic acid obtained in the fermentation broth is usually near about 100 grams per liter of glutamic acid present in the fermentation media. Now we will see the recovery of the glutamic acid. So, usually the separation or the purification steps in the in the glutamic acid produced in the fermentation media, it can be carried out by centrifugation, filtration, crystallization, ion exchange, chromatography, electrodialysis, solvent extractions, then decolorization, and then evaporation. So, you know that after the fermentation is completed, you have to undergo the centrifugation in order to remove the that is the bacterial cells, and then you this filtration is undergone the crystallization steps, and in order to have the purified form of glutamic acid, you have to undergo the ion exchange, electrodialysis, solvent extraction, etc. So, the glutamic acid crystals is added to the sodium hydroxide solution in order to get the monosodium glutamate, and this monosodium glutamate is more soluble in water, less likely absorb moisture, and has the strong umami taste. Okay. And this monosodium glutamate it is cleansed by using the active carbon. This is the active carbon which is used, which has many micro holes on their surface. So, by means of adsorption, usually the monosodium glutamate is purified. The clean monosodium glutamate solution is concentrated by heating, and then you will get the crystals, and these crystals produced are dried in hot air in a closed system, and then it is packaged, and then it is ready to sold. So, this is your pure monosodium glutamate, and this is the charcoal which is used for the adsorption purification. Thank you. Hope you have enjoyed this session.